I'm Marco Toffoli. I'm a clinical research fellow here at the Royal Free Hospital. I'm doing a PhD in neurosciences under Professor Schapira, and I'm working on Rapsodi, which stands for Remote Assessment of Parkinsonism Supporting Ongoing Development of Interventions. You just need to remember the Rapsodi short name. So I would like to explain to you shortly why we're doing the study, why it's incredibly important that you did take part or will take part if you didn't do it yet, and then where we hope to arrive with our study. So just a quick slide of background. It has already been said that a part of people that carry GBA mutations, the ones that cause Gaucher disease, will develop Parkinson's disease. They have an increased risk. And this risk, it's very difficult to estimate, but it is around 5 to 30% of people with GBA mutations will develop Parkinson. Of course, it's quite a wide range of risk, and it changes according to the study you take into account, but there is an increase. But it's important to note that not every person that has a GBA mutation will develop Parkinson in his lifetime. And so that's one thing that is very important. We also know that around 10% of people that have Parkinson's disease carry GBA mutations. There are two questions that brought us to design the study. The first one is it possible to spot the GBA carriers that will develop Parkinson before they do, so that we can maybe use some therapies to slow down the disease or even to prevent it from developing? And the second question is there an easy way to spot those people that have Parkinson that also have GBA mutations? Because we say that we will see later this afternoon that some therapies might be useful to these people that carry GBA mutations and have Parkinson, of course in the setting of clinical trials. But it's not that easy to sequence the gene and to just look at a Parkinson patient and say, well, I think you have GBA mutations. So that's the two questions that brought us to develop this study. Regarding the first one, how can we understand how to predict who among the GBA carriers will develop Parkinson? The idea is simple. You take a lot of GBA carriers, you follow them up over time, and you find out who develops Parkinson. You then go back at the baseline, and you see what were the characteristics of these people, and then you can say, well, if you have this characteristic, then you have a higher risk of developing Parkinson. So if we take all the GBA carriers here, at baseline, we assess them for a lot of non-motor symptoms for Parkinson's disease and a lot of other things. We then follow them up, and we find, let's say, 2 that develop Parkinson. And then we can go back at the baseline and say, well, these 2 people had a decreased sense of smell. They were taller than the others, whatever. And so we can say, if you are taller than the others, then you will develop Parkinson. I'm not saying if you're taller, you will develop Parkinson, of course, but that you get the point. And we will follow them up even further. We have, the plan is to follow people up for up to 25 years. And then we can find maybe an algorithm, a way to predict the risk of developing Parkinson among people that carry GBA mutations. We tried to do so, well, we did so in the past. I didn't do that personally, but the team that works on GBA and Parkinson here did. And they selected some people that carry GBA mutations and assessed them in person. They published already 2 very good papers that were, I would say, game-changing in the setting of GBA and Parkinson, and 2 more hopefully will come up soon. But there were some problems. The first one is that people, if you try to assess the same person for 6-8 years and ask them to come every other year, they might get bored after a while. And so there were a lot of dropouts, a lot of people that didn't come back. They were busy, they forgot. So that was the first problem, that a lot of people dropped out of the study. And the second problem is that despite all the effort, it was very hard to reach a big number of GBA carriers because they were all around the UK. there is a huge cohort here in London, but of course there's a lot more out of London. So that was another problem. And regarding the second question, so are we able to spot any difference among Parkinson patients that can suggest that they carry the GBA mutation? Well, you just take a lot of Parkinson people and you genotype them, and then you find some that carry the mutation. And so then you can just see what are the differences. Problem is, we say that only 10% of Parkinson people carry GBA mutations. So in order to find 10, you need to sequence 100 patients. In order to find 100, you need to sequence 1,000. It's very hard to reach 1,000 people if you want to see them all in person. Well, Stephen Malling actually got the brilliant idea to overcome these problems by using technology. We know that almost every house now has a computer, and almost, well, not everybody, but a lot of people know how to use a computer, and they're all interconnected. So there might be a way, there is actually, we're using it, to assess people from home. So we can reach a lot more people. It's a lot more comfortable for them because they just sit on their sofa and they play some games on the computer. And we are actually able to reach the numbers that we need to do a meaningful study. How does the study work? For those of you that didn't do it yet, we have an online portal. So you log into the internet from your computer, you click on a link, and you are prompted to do some questionnaires. Basically, you just answer to some multiple choice questions. You do a tap test, you press on a keyboard as fast as you can, trying to hit the right keys, of course. And then you do some more questionnaires, and then you do a memory test. The memory test is like a board game, in a sense. It's just they show you some images, and you need to try and remember them. You see some numbers, you need to try and remember them. And this is to assess some fields of cognition that we think might point out that you might develop Parkinson or you have a particular form of Parkinson. And then we send you a booklet with some smells. You smell them and you try to guess what they are. You send the booklets back together with a saliva sample that you can collect in the tube that we provide. And then we sequence the gene through the saliva that you sent us. And then from that point on, we ask you to repeat the assessment every year, just the part that is done at home without the booklets and the saliva kit. And this is to follow people up over time, see who develops Parkinson, if they do, and to see how the progression of Parkinson goes in people that already have Parkinson. So it's really easy, really quick to do, and it's giving good results so far. This is the kit that we send to people after they complete the first bit. This is the saliva collection kit. You just open it, spit in it, and then send it back. These are the smell booklets, where you scratch a small brown square, you smell it, and try to guess it. And then you send everything back in this yellow envelope. But I'm sure that most of you already know that. Who can take part in Rapsodi? Gaucher disease patients, Parkinson's disease patients, and we got very good response, I would say, from both of them. And then people that know already, for any reason, that they carry the GBA mutation, and family members of Gaucher disease patients. That's very important. That is very important because if you are a relative of someone with Gaucher disease, you have a chance of carrying GBA mutations. And so by recruiting family members, we hope to increase the number of GBA carriers. That's really, really important. And if you know any relative of a Gaucher disease patient that might be interested in taking part, we are more than happy to have them in the study. Also, spouses of people with Parkinson's disease can take part because, of course, we need controls in our study. Spouses are supposed to be healthy, not to have Parkinson's disease, and so we can, and they will be matched more or less for age, and so they are very valuable controls to us. How you can take part if you didn't do that yet, you can go to the study. We have flyers with the link. Or you can just send an email to us. In the flyers, we have the email address of Soraya and me. Or you can just leave us your email address here today, or take part through the computers that we will have in the back of the room later today. The recruitment goals for Rapsodi are 800 total participants with at least 100 GBA carriers and 100 GBA plus Parkinson's disease patients. How are we doing so far? The total number of people that we recruited is almost what we are hoping to achieve. We are getting there with the GBA carriers, although it's slowing down. So again, family members are very important. We're doing our best, but we can improve the GBA plus Parkinson's disease patients. And that's due to what I was explaining to you. We need to reach a huge number of Parkinson's people to get to 100 GBA plus Parkinson patients. And if you do the math, actually, it's 1,000 Parkinson patients. And so, again, if you have Gaucher disease, know that you carry the GBA gene, and you have a relative with Parkinson's disease, this person will be really valuable to the study. So, goes without saying, you can talk to them about Rapsodi. Just as a final slide, a word about genotyping. GBA gene is a very difficult gene to genotype. I won't go into detail, but there is no NHS approved test because it's very difficult to sequence. And if you do it through an accredited lab, it's very expensive because they need to look at small bits of gene one at a time, because there are other pseudogenes that are very similar and they can mess up everything with the sequencing. So we are using now a technique that we think is very innovative. It was invented in Oxford by Oxford Nanopore. It's called a nanopore. And basically you put the DNA extracted from saliva of up to 96 people inside this small machine that you can plug into a computer via USB. And you get the full gene sequencing of the GBA gene. So that's unimaginable. Just 10 years ago, it was impossible to think of something like that. And you get it for 10 pounds per person, maybe. So a lot less than what it would cost to do the standard sequencing technique. Of course, we are still optimizing it. So it takes a while. We are still learning. And this means that, of course, we confirm all our results with the standardized techniques before giving it out to anybody that wants to know that. But that means that it takes a while for us to sequence samples. And that's to answer to any question that might arise regarding when is my sequencing results coming back. It will take a while, but that's why, because we are trying to develop something that might change the way GBA is sequenced, and so we want to be completely sure and to have everything in place before we reach out to people. And that's everything. Thank you.